Hello everyone, and welcome to today's presentation. I'm excited to introduce the topic, BCHM 315, Pathways of Amino Acid Degradation. Today, we'll explore how amino acids are broken down in the body, covering key pathways and mechanisms. We'll also look at how these processes are interconnected and their importance in metabolism. Let's get started. The pathways of amino acid catabolism, taken together, normally account for only about 10 to 15% of the human body's energy production. This means that these pathways are not nearly as active as glycolysis and fatty acid oxidation, which are the primary sources of energy. The flux, or flow, through these amino acid breakdown routes can vary greatly depending on the body's needs. If the body requires more for biosynthetic processes, less amino acid is directed towards energy production, and vice versa. Interestingly, despite there being 20 different amino acids with their own specific pathways, all of these catabolic routes converge into just 6 major products. These products then enter the citric acid cycle, which is central to energy production. From this point, the carbon skeletons of amino acids can be diverted into different metabolic pathways, either towards glucogenesis to produce glucose, ketogenesis to generate ketone bodies, or they can be fully oxidized to carbon dioxide and water for energy. This overall process highlights how amino acids contribute to energy metabolism, but also how their primary role is in supporting biosynthesis and maintaining metabolic flexibility. Understanding these pathways helps us appreciate the complexity and efficiency of our metabolic network. Let's take a closer look at how amino acids contribute to energy production through the citric acid cycle. Essentially, the carbon skeletons of 7 amino acids are ultimately broken down into acetyl-CoA, which then enters the cycle to generate energy. Now, 5 amino acids are converted into alpha-ketoglutarate, and 4 are transformed into succinyl-CoA. Additionally, 2 amino acids become fumarate, and another 2 are converted into oxaloacetate. These are all key intermediates within the cycle, highlighting how amino acids can feed into different points here. Furthermore, parts or entire carbon skeletons of 6 amino acids are converted into pyruvate. From there, pyruvate has 2 possible paths. It can be turned into acetyl-CoA or into oxaloacetate, depending on the body's needs. Later in our discussion, we'll look at flow diagrams that map out these individual pathways for all 20 amino acids. Each pathway shows a specific entry point into the citric acid cycle, with the carbon atoms entering the cycle highlighted in color for clarity. It's also important to note that some amino acids can appear more than once in these pathways, reflecting that different parts of their skeletons can have different destinies within metabolism. Rather than going through every step of each pathway, I want to focus on some key enzymatic reactions that are particularly interesting because of their mechanisms or their significance in medicine. This overview helps us understand how amino acids are versatile contributors to energy metabolism, connecting protein catabolism directly with the citric acid cycle's function. Let's take a closer look at the 7 amino acids that are either fully or partially degraded into acetoacetyl-CoA and or acetyl-CoA. These are phenylalanine, tyrosine, isoleucine, leucine, tryptophan, threonine, and lysine. What's important about these amino acids is that they can produce ketone bodies in the liver. Specifically, acetoacetyl-CoA gets converted into acetoacetate, which then can turn into other ketone bodies like acetone and beta-hydroxybutyrate. These are what we call the ketogenic amino acids. Their ability to generate ketone bodies becomes especially noticeable in certain conditions, such as uncontrolled diabetes mellitus. In such cases, the liver ramps up production of ketone bodies, not just from fatty acids, but also from these ketogenic amino acids. This can lead to a significant increase in ketone levels in the blood. Additionally, ketone bodies aren't just produced in the liver. They can also serve as an alternative fuel source for the brain, especially during starvation. When glucose is scarce, the brain can utilize ketone bodies for energy, helping to sustain vital functions. It's also worth noting that some amino acids are both glucogenic and ketogenic, meaning they can contribute to glucose production and ketone body formation. This dual capability plays a crucial role in how our body manages energy supply during different metabolic states, linking back to the overall theme of energy metabolism and balance. Let's take a closer look at how amino acids contribute to glucose production. Some amino acids are degraded into key intermediates like pyruvate, alpha-ketoglutarate, succinyl-CoA, fumarate, and oxaloacetate. These can then be used to synthesize glucose and glycogen through various pathways. We call these the glucogenic amino acids. It's important to note that the boundary between ketogenic and glucogenic amino acids isn't always clear-cut. In fact, 5 amino acids, tryptophan, phenylalanine, tyrosine, threonine, and isoleucine, are both ketogenic and glucogenic, meaning they can contribute to either pathway depending on the metabolic context. Interestingly, all amino acids except lysine and leucine can potentially support gluconeogenesis, which is vital during fasting or starvation when glucose needs to be maintained. This highlights how crucial amino acid breakdown is for survival, especially in animals on high-protein diets or during periods of fasting. Focusing on leucine, it's unique because it's purely ketogenic. It doesn't contribute to glucose formation, but instead feeds into ketone body production. During starvation, the breakdown of leucine significantly contributes to ketosis, providing an alternative energy source when glucose is scarce. So, in summary, understanding the balance and roles of these amino acids helps us see how the body adapts to different nutritional states, ensuring energy supply and metabolic stability. Now, let's take a closer look at glucogenic and ketogenic amino acids. These are 2 important categories based on how amino acids are used in metabolism. Glucogenic amino acids are those that can be converted into glucose through a process called gluconeogenesis. This means they can help maintain blood sugar levels, especially during fasting or intense exercise. On the other hand, ketogenic amino acids are those that are broken down into compounds that can be converted into ketone bodies, providing an alternative energy source when glucose is scarce. Some amino acids are purely glucogenic, some are purely ketogenic, and others are both. This dual capability is crucial because it shows how amino acids contribute to different energy pathways depending on the body's needs. Understanding these differences helps us appreciate how protein metabolism supports overall energy balance, especially during states of fasting or prolonged activity. So, connecting this back to the big picture, knowing whether an amino acid is glucogenic or ketogenic allows us to understand how the body adapts to various metabolic demands, ensuring a continuous energy supply. The structural diversity of amino acids is really reflected in the variety of reaction types we see during their breakdown pathways. To start understanding these, we first need to look at some key classes of reactions that show up repeatedly, along with the enzyme cofactors involved in them. We've already discussed one important class, transamination reactions, which require pyridoxal phosphate. Now, another common reaction in amino acid catabolism is one-carbon transfer. These reactions typically involve one of 3 main cofactors: biotin, tetrahydrofolate, or S-adenosyl methionine. Each of these cofactors transfers 1 carbon groups, but in different oxidation states. For example, biotin handles carbon in its most oxidized form, which is Co2. Tetrahydrofolate is more versatile, transferring 1 carbon groups that are in intermediate oxidation states, sometimes as methyl groups. And S-adenosyl methionine is the most reduced form, transferring methyl groups. These latter 2 cofactors, tetrahydrofolate and S-adenosyl methionine, are especially important not just in amino acid breakdown, but also in nucleotide metabolism. So, understanding these cofactors helps us see how interconnected these pathways are and highlights their significance in overall metabolism. Let's talk about the anemia related to vitamin B12 deficiency, which is known as megaloblastic anemia. This condition occurs because there's a decline in the production of mature red blood cells, or erythrocytes. In the bone marrow, you'll notice the appearance of immature precursor cells called megaloblasts. In the bloodstream, these developing cells are gradually replaced by fewer, abnormally large erythrocytes, known as macrocytes. The core issue here stems from a defect in erythrocyte development, which happens because of the depletion of a specific compound called N5, N10-methylnitetrahydrofolate. This molecule is essential for synthesizing thymidine nucleotides, which are vital for DNA replication. Interestingly, folate deficiency, where all forms of tetrahydrofolate are depleted, can cause a similar type of anemia for similar reasons. Both B12 deficiency and folate deficiency impair DNA synthesis in rapidly dividing cells like those in the bone marrow. Now, the good news is that symptoms of this kind of anemia can often be alleviated by supplementing with either vitamin B12 or folate. So, understanding these pathways highlights how interconnected vitamin B12 and folate are in maintaining healthy blood cell production. However, it is important to remember that treating pernicious anemia with just folate supplements can be dangerous. This is because the neurological symptoms caused by B12 deficiency tend to worsen if B12 isn't also provided. These neurological issues don't result from problems in the methionine synthase reaction itself, but rather from impaired methylmalonyl-CoA mutase activity. When this enzyme isn't working properly, it leads to an accumulation of unusual, odd-number fatty acids in neuronal membranes, which can cause significant neurological damage. That's why, when treating anemia related to folate deficiency, we usually administer both folate and vitamin B12 together. This approach continues at least until we're certain about the underlying metabolic cause of the anemia. Early diagnosis of B12 deficiency is crucial because some of its associated neurological conditions can become irreversible if not addressed promptly. And as we move into understanding enzyme cofactors, it's clear how vital these molecules are in amino acid catabolism, supporting the overall metabolic health and preventing such deficiencies. Let's take a closer look at some enzyme cofactors that play crucial roles in one-carbon transfer reactions. These cofactors are essential because they help enzymes facilitate the transfer of one-carbon units, which are vital in various metabolic pathways. When we talk about one-carbon transfer, we're often referring to processes like amino acid synthesis, nucleotide biosynthesis, and methylation reactions. The most important cofactors in these processes include molecules like tetrahydrofolate, which acts as a carrier for one-carbon units, and vitamin B12, which is involved in methyl transfer reactions. Understanding these cofactors helps us see how interconnected and finely tuned these metabolic pathways are. They essentially act as helpers, ensuring that the transfer of one-carbon units happens efficiently and accurately. This is fundamental to maintaining proper cellular function and overall metabolic health, which ties back to our broader theme of metabolic regulation and enzyme function. Now, let's take a closer look at how 6 specific amino acids are degraded into pyruvate, which is a key metabolic intermediate. The amino acids we're focusing on are alanine, tryptophan, cysteine, serine, glycine, and threonine. These can be converted in whole or in part into pyruvate, linking amino acid catabolism directly to energy production and gluconeogenesis. Starting with alanine, it directly yields pyruvate through a process called transamination with alpha-ketoglutarate. This is an important pathway because alanine can serve as a shuttle of nitrogen and carbon between tissues and the liver. Tryptophan is a bit more complex. Its side chain is cleaved during degradation, ultimately producing alanine, which then becomes pyruvate. So, in essence, tryptophan's breakdown contributes to pyruvate pools indirectly. Cysteine is converted into pyruvate through a 2-step process. First, the sulfur atom is removed, and then a transamination occurs, transforming it into pyruvate. This highlights the versatility of amino acid catabolism pathways. Serine, on the other hand, is converted directly into pyruvate by an enzyme called serine dehydratase. This reaction involves removing both the beta-hydroxyl and the alpha-amino groups in a single step, which depends on a cofactor called pyridoxal phosphate. All of these pathways demonstrate how amino acids can feed into central metabolic pathways, either for energy production via the citric acid cycle or for glucose synthesis through gluconeogenesis. This interconnectedness is essential for maintaining metabolic flexibility and energy balance in the body. Glycine, as you might recall, can be broken down through 3 different pathways, but interestingly, only one of these pathways ultimately produces pyruvate. Let's focus on that one first. In this pathway, glycine is converted into serine through an enzymatic process that adds a hydroxymethyl group. This reaction is catalyzed by serine hydroxymethyltransferase, and it requires 2 important coenzymes, tetrahydrofolate and pyridoxal phosphate. Once serine is formed, it can then be converted into pyruvate, completing its degradation pathway. Now, moving on to the second pathway, which is actually more common in animals. Here, glycine undergoes an oxidative cleavage process, where it's broken down into carbon dioxide, ammonium, and a methylene group. That's a CH2 group. This reaction is reversible and is catalyzed by the glycine cleavage enzyme, also known as glycine synthase. This enzyme also needs tetrahydrofolate, which plays a key role by accepting the methylene group during this process. It's important to note that in this oxidative cleavage pathway, the 2 carbons originally present in glycine don't go into the citric acid cycle. Instead, 1 carbon is released as CO2, and the other becomes part of a molecule called N5, N10-methylnitrohydrofolate. This molecule is important because it donates 1 carbon groups in various biosynthetic pathways, linking amino acid degradation to broader metabolic processes. So, as we see, glycine degradation is versatile, connecting amino acid metabolism with energy production and biosynthesis, which is a key theme as we explore how amino acids are broken down and utilized in the body. This second pathway for glycine degradation appears to be especially important in mammals. When humans have serious defects in the activity of the glycine cleavage enzyme, they develop a condition called non-ketotic hyperglycemia. This condition is characterized by elevated levels of glycine in the serum, which can lead to severe intellectual disability and even death in very early childhood. Now, at high levels, glycine acts as an inhibitory neurotransmitter, and that could help explain some of the neurological symptoms we see in this disease. But perhaps more critically, the excess glycine increases the levels of a compound called 2-amino-3-ketabutyrate. This is an unstable intermediate in the mitochondrial degradation of threonine. What happens next is quite concerning. 2-amino-3-ketabutyrate can spontaneously decarboxylate to form aminocetone, which is toxic. This metabolite is then readily converted into methylglyoxal, a highly reactive molecule that can modify proteins and DNA, leading to cellular damage. So overall, this pathway highlights how disruptions in glycine degradation can have serious neurological and cellular consequences, linking to the broader theme of amino acid metabolism and its importance in maintaining health. In the third and final pathway of glycine degradation, the acyryl glycine molecule becomes a substrate for the enzyme deamino acid oxidase. This enzyme specifically acts on glycine, converting it into glyoxylate, which then serves as an alternative substrate for lactate dehydrogenase. Next, glyoxylate undergoes an NAD+ dependent oxidation reaction, resulting in the formation of oxalate. This pathway highlights how glycine can be broken down through different routes, ultimately connecting amino acid metabolism to other crucial metabolic intermediates like oxalate. It's also interesting to note that 6 amino acids, in general, are degraded into pyruvate, emphasizing the interconnected nature of amino acid and carbohydrate metabolism. This pathway underscores the diverse roles amino acids play in energy production and metabolic regulation within the body. Let's take a closer look at the catabolic pathways for some of these amino acids: alanine, tryptophan, cysteine, serine, glycine, and threonine. These pathways are essential because they show us how our body breaks down amino acids to produce energy or other metabolites. For example, alanine is involved in the glucosalanine cycle, which helps in maintaining blood sugar levels during fasting. Tryptophan, on the other hand, can be degraded into molecules that serve as precursors for neurotransmitters like serotonin. Cysteine breakdown involves pathways that contribute to the synthesis of important compounds like glutathione, a key antioxidant. Serine and glycine are interconnected in their catabolism, often feeding into one-carbon metabolism, which is vital for nucleotide synthesis and methylation reactions. Threonine, meanwhile, can be broken down into compounds that enter energy-generating pathways. Overall, understanding these pathways highlights how amino acids are not just building blocks for proteins, but also crucial players in metabolic regulation and energy production. Let's take a closer look at how 7 different amino acids are broken down into key metabolic intermediates. Specifically, the portions of their carbon skeletons can be converted into acetyl-CoA or acetoacetyl-CoA. Now, remember, acetoacetyl-CoA can also be converted into acetyl-CoA, so there's a bit of flexibility there. What's interesting is that some of the final steps in the breakdown pathways for leucine, lysine, and tryptophan resemble steps we see in fatty acid oxidation. This highlights how interconnected these metabolic pathways are. Additionally, threonine, although it primarily follows a different route, can produce some acetyl-CoA through a minor pathway. Overall, this slide emphasizes that these amino acids contribute to energy production by feeding into central metabolic pathways, especially those involving acetyl-CoA. Let's take a closer look at the degradative pathways of some of these amino acids, specifically 2 that deserve a bit of special attention. First, tryptophan. It's quite unique because its breakdown process is actually the most complex among all amino acids in animal tissues. When tryptophan is metabolized, part of its structure, specifically 4 of its carbons, is converted into acetyl-CoA through a molecule called acetoacetyl-CoA. This pathway isn't just about energy production. It's also significant because some intermediates in tryptophan catabolism serve as precursors for other vital biomolecules. For instance, nicotinate, which is a precursor for NAD and NADP, key molecules in cellular redox reactions. Additionally, tryptophan catabolism leads to the formation of serotonin, which is well known as a neurotransmitter in vertebrates, influencing mood and other functions. And in plants, one of the intermediates, indole acetate, acts as a growth factor, highlighting the diverse roles of these metabolic pathways across different organisms. Overall, understanding these pathways helps us see how amino acids are not just building blocks for proteins, but also crucial for producing molecules essential for various biological functions. And recognizing the complexity of tryptophan breakdown underscores its importance in metabolism and cell regulation. Now, let's look at how oxidation products of tyrosine, which both contain 9 carbons, are broken down into 2 key fragments. Both of these fragments can then enter the citric acid cycle, which is central to cellular energy production. 4 of the 9 carbons from these fragments are converted into free acetoacetate. This acetoacetate is then transformed into acetoacetyl-CoA, which ultimately becomes acetyl-CoA, a crucial molecule in energy metabolism. The second 4-carbon fragment is recovered as fumarate, another intermediate in the citric acid cycle. In total, 8 of the 9 carbons from these 2 amino acids successfully enter the cycle, contributing to energy production. The remaining carbon is lost as carbon dioxide, highlighting how the body efficiently manages amino acid degradation. It's also interesting to note that phenylalanine, after being hydroxylated to tyrosine, serves as a precursor for several important compounds. It's the starting point for dopamine, a key neurotransmitter, as well as norepinephrine and epinephrine, which are hormones released by the adrenal medulla. Additionally, tyrosine is the precursor for melanin, the pigment responsible for the color of our skin and hair. So, from a single amino acid, the body produces a variety of critical molecules involved in neurochemical signaling, hormone regulation, and pigmentation. Lastly, it's worth mentioning that 7 amino acids are degraded into acetyl-CoA, emphasizing the central role of this molecule in connecting amino acid metabolism to the citric acid cycle and overall energy production. Now, let's take a closer look at how certain amino acids are broken down in the body. Specifically, 7 amino acids are degraded into acetyl-CoA. This is a crucial point because acetyl-CoA plays a central role in energy production, feeding directly into the Krebs cycle. When these amino acids are converted into acetyl-CoA, they can then be used to generate ATP, which is our cell's main energy currency. This process highlights the versatility of amino acids. They can serve not only as building blocks for proteins, but also as sources of energy when needed. Understanding this pathway helps us see how the body efficiently manages different nutrients and maintains energy balance, fitting into the bigger picture of metabolism we're exploring in this presentation. Given that many amino acids serve as neurotransmitters, precursors, or even antagonists of neurotransmitters, it's not surprising that genetic defects in amino acid metabolism can lead to issues with neural development and intellectual disabilities. When these metabolic pathways are disrupted, specific intermediates tend to accumulate in the body. For example, in the case of phenylketonuria, or PKU, a genetic defect affects the enzyme phenylalanine hydroxylase, which is the first enzyme involved in breaking down phenylalanine. This defect causes phenylalanine to build up in the blood, leading to hyperphenylalanemia. So, this slide highlights how genetic issues in amino acid catabolism can have significant neurological consequences, especially when specific enzymes are defective. Phenylalanine hydroxylase, which is also known as phenylalanine 4-monooxygenase, belongs to a broader class of enzymes called mixed-function oxygenases. These enzymes are fascinating because they catalyze a unique reaction. They hydroxylate a substrate by using one oxygen atom from O2, while the other oxygen atom is reduced to form water. Now, for phenylalanine hydroxylase to do its job, it needs a special cofactor called tetrahydrobiopterin. This cofactor plays a crucial role by carrying electrons from NADPH to oxygen, enabling the hydroxylation process. During this transfer, tetrahydrobiopterin gets oxidized to dihydrobiopterin. But the cycle doesn't end there. The enzyme dihydrobiopterin reductase steps in to reduce dihydrobiopterin back to tetrahydrobiopterin, and this reaction also requires NADPH. This entire process is essential for proper phenylalanine metabolism. It's important to understand that in some people, this pathway is genetically defective. When that happens, phenylalanine isn't properly broken down, which can lead to metabolic issues like phenylketonuria. So this slide really highlights how the precise function of enzymes like phenylalanine hydroxylase is vital not just for normal metabolism, but also for overall health. In individuals with PKU, we see that a secondary pathway of phenylalanine metabolism comes into play, and this pathway isn't usually active in healthy people. Here, phenylalanine undergoes transamination with pyruvate, which produces phenylpyruvate. As a result, phenylalanine and phenylpyruvate tend to build up in the blood and tissues, and they are eventually excreted in the urine. That's actually where the name phenylketonuria comes from, referring to the phenylketones like phenylpyruvate found in the urine. Much of this phenylpyruvate doesn't just stay as it is. It can be decarboxylated into phenylacetate or reduced to phenylactate. And this is quite significant because phenylacetate gives urine a distinctive honey-like odor, which nurses have traditionally used to help detect PKU in infants. Now, the accumulation of phenylalanine and its metabolites early in life can seriously impair brain development. This leads to severe intellectual disabilities if left untreated. One reason this happens is that excess phenylalanine competes with other amino acids for transport across the blood-brain barrier, creating a shortage of essential metabolites needed for normal brain development. It's important to remember that phenylalanine catabolism is genetically defective in some people, which is the root cause of PKU. This genetic defect prevents proper breakdown of phenylalanine, leading to these metabolic issues we've discussed. Phenylketonuria, or PKU, was actually one of the first inheritable metabolic defects discovered in humans. It's a condition that highlights how genetics can directly influence our biochemistry. When we catch this condition early in infancy, we can often prevent serious outcomes like intellectual disability through strict dietary control. This means carefully managing the intake of certain amino acids, like phenylalanine. The key point here is that early detection and intervention are crucial. This ties into the overall theme of how understanding genetic metabolic disorders can help us develop effective treatments and preventative strategies. Now, let's look at why this happens. Specifically, how phenylalanine catabolism is genetically defective in some people. The diet for individuals with phenylketonuria must supply only enough phenylalanine and tyrosine to meet the body's needs for protein synthesis. This means that consumption of protein-rich foods needs to be carefully limited. Natural proteins, like casein found in milk, must first be hydrolyzed, that is, broken down, and much of the phenylalanine removed to create an appropriate diet, especially during childhood. It's also important to be aware that foods sweetened with aspartame, which is a dipeptide of aspartate and the methyl ester of phenylalanine, can pose a risk. Because of their composition, these foods carry warnings for individuals on phenylalanine-controlled diets. Lastly, I want to highlight that phenylalanine catabolism, the body's process of breaking down phenylalanine, is genetically defective in some people. This genetic defect leads to a buildup of phenylalanine, which can cause serious health issues if not managed properly. So, understanding these points is crucial for managing phenylketonuria effectively. Following a strict diet can be quite challenging to do perfectly over a lifetime, and because of that, it often doesn't fully eliminate neurological symptoms associated with phenylketonuria. To address this, a new enzyme substitution treatment has been developed. This involves modifying an enzyme called phenylalanine ammonia lyase with polyethylene glycol, or PEG, which helps it stay stable longer in the body. It's then injected under the skin and works to break down phenylalanine from the proteins we eat, allowing people to have a slightly less restricted diet. This enzyme isn't something new. It's derived from plants, bacteria, yeast, and fungi. Normally, phenylalanine ammonia lyase plays a role in plants and microbes by helping produce compounds called polyphenols, like flavonoids. Its main job is to convert phenylalanine into transaminamic acid and ammonia, which are harmless. The small amount of ammonia produced isn't toxic, so it's a safe process. This treatment was approved for use in patients back in 2018. And since then, researchers have been studying its long-term effects to better understand its benefits and safety. Now, it's important to remember that phenylalanine metabolism is genetically defective in some people, which is what causes phenylketonuria. This genetic defect makes it difficult for the body to process phenylalanine properly, leading to the issues we've been discussing. Phenylketonuria, or PKU, can also result from a defect in the enzyme responsible for regenerating tetrahydrobiopterin, which is a crucial cofactor. When this enzyme isn't working properly, the treatment becomes more complex than simply restricting phenylalanine and tyrosine intake. That's because tetrahydrobiopterin is not only involved in phenylalanine metabolism, but is also essential for producing important neurotransmitter precursors like L34-dihydroxyphenylalanine, or L-DOPA, and 5-hydroxytryptophan. These are the precursors to norepinephrine and serotonin, which are vital for brain function and mood regulation. In cases where this enzyme defect is the cause, we need to supply these precursors directly through the diet, along with tetrahydrobiopterin itself, to support both metabolic and neurotransmitter functions. This highlights the importance of screening newborns for genetic diseases like PKU. Early detection can be highly cost-effective because it allows for timely intervention, preventing severe neurological damage and improving quality of life. So overall, understanding these different causes of PKU helps us tailor treatments and emphasizes the value of early screening in newborns. Now, let's talk about the tests used for detecting PKU. These tests are no longer relying on the old method of checking urine odor, which was less reliable and more invasive. Instead, they are relatively inexpensive, making them accessible for widespread screening. The key benefit here is that early detection and treatment in infants, who are affected in about 8 to 10 cases per 100,000 newborns, can actually save millions of dollars each year in healthcare costs down the line. But beyond the financial savings, what really matters is the emotional impact. Early detection through these simple tests can help avoid a great deal of emotional trauma for families. Imagine the relief of knowing early on and being able to intervene, rather than facing uncertainty or the consequences of delayed diagnosis. This is a perfect example of how advancements in screening not only improve health outcomes, but also significantly reduce the emotional burden on families, reinforcing the overall importance of early detection in our healthcare approach. Let's talk about another inheritable disease related to phenylalanine catabolism, called alkaptonuria. The defective enzyme here is homogentisate dioxygenase. Compared to PKU, it's less serious and usually causes fewer problems. However, it does lead to the excretion of large amounts of homogentisate, and when this oxidizes, it turns the urine black, something quite distinctive. People with alkaptonuria are also more likely to develop a type of arthritis, which adds to the health issues they face. Now, this condition is actually of significant historical interest. In the early 1900s, Archibald Garrod discovered that it's inherited and traced the cause back to the absence of a single enzyme. This was a groundbreaking insight because Garrod was the first to link an inheritable trait directly to an enzyme. This connection laid important groundwork for our current understanding of genes and the pathways of genetic information, which we'll explore further in part III. So, in essence, alkaptonuria not only teaches us about metabolic diseases, but also helped shape the foundation of modern genetics. Now, let's look at how the carbon skeletons of 5 specific amino acids, proline, glutamate, glutamine, arginine, and histidine, enter the citric acid cycle. They all feed into the cycle as alpha-ketoglutarate, which is a key intermediate. Focusing first on proline, glutamate, and glutamine, these amino acids have 5 carbon skeletons. That's important because it means they can be directly funneled into the cycle after some transformations. Starting with proline, its cyclic structure is opened through oxidation. The oxidation occurs at the carbon farthest from the carboxyl group, resulting in the formation of a Skiff base. This Skiff base is then hydrolyzed, breaking it down into a linear semialdehyde, specifically glutamate gamma semialdehyde. This intermediate can then be further oxidized at the same carbon to produce glutamate. Glutamine, on the other hand, is converted into glutamate through the action of glutaminase or other enzyme reactions that transfer its amide nitrogen to an acceptor. This process is vital for nitrogen metabolism. Finally, through transamination or deamination of glutamate, we produce alpha-ketoglutarate, which then enters the citric acid cycle. So, in summary, these 5 amino acids are converted into alpha-ketoglutarate through a series of enzymatic reactions, linking amino acid metabolism directly to the citric acid cycle and overall energy production. Let's take a closer look at arginine and histidine. Both of these amino acids have a unique structure with 5 adjacent carbons and a 6th carbon that's attached through a nitrogen atom. This structural detail makes their breakdown pathways a bit more complex compared to amino acids like proline or glutamine. For arginine, the process begins in the urea cycle, where it's converted into a 5-carbon skeleton called ornithine. From there, ornithine undergoes transamination to produce glutamate gamma semialdehyde. Histidine's conversion to glutamate is even more intricate, involving multiple steps. One key point is that during this process, the extra carbon is removed, and this step requires tetrahydrofolate as a cofactor. Understanding these pathways highlights how the structure of amino acids influences their catabolic routes. This knowledge ties back to our overall theme of amino acid metabolism and how structural nuances impact metabolic complexity. Let's take a closer look at how the carbon skeletons of these amino acids, methionine, isoleucine, threonine, and valine, are degraded. They all feed into the citric acid cycle through a common intermediate succinyl-CoA. Starting with methionine, it donates its methyl group to acceptors via S-adenosyl methionine. The remaining 3 carbons from methionine are converted into propionyl-CoA, which then becomes succinyl-CoA. Isoleucine's pathway begins with transamination, followed by oxidative decarboxylation of the resulting alpha-keto acid. Its 5-carbon skeleton is then further broken down into acetyl-CoA and propionyl-CoA, both of which can enter energy-generating pathways. Valine also undergoes transamination and decarboxylation, and through a series of oxidation reactions, its 4 remaining carbons are converted into propionyl-CoA. Interestingly, parts of the degradation pathways for valine and isoleucine are quite similar to steps involved in fatty acid breakdown. Turning to threonine, in human tissues, it's primarily converted into propionyl-CoA through 2 steps. The first step resembles the reaction catalyzed by serine dehydratase, and there's evidence that serine and threonine dehydratases might actually be the same enzyme. Overall, what's fascinating is that these 4 amino acids, methionine, isoleucine, valine, and threonine, are all funneled into succinyl-CoA, integrating amino acid catabolism with the citric acid cycle and energy production. Now, let's look at how propionyl-CoA, which is derived from 3 specific amino acids, is converted into succinyl-CoA, an important intermediate in the Krebs cycle. The process involves a series of steps. First, propionyl-CoA undergoes carboxylation to form methylmalonyl-CoA. Then, this methylmalonyl-CoA is epimerized, meaning its stereochemistry is changed, to prepare it for the final step. The last step involves converting methylmalonyl-CoA into succinyl-CoA, and this reaction depends on the enzyme methylmalonyl-CoA mutase, which requires vitamin B12 as a coenzyme. This pathway is crucial because it links amino acid metabolism to energy production. However, in a rare genetic disorder called methylmalonic acidemia, this process is disrupted because methylmalonyl-CoA mutase is missing or not functioning properly. As a result, methylmalonyl-CoA builds up, leading to serious metabolic problems. This highlights how essential proper enzyme function is for maintaining metabolic balance and overall health. This slide doesn't contain enough information. That's why we're moving to the next slide. Now, let's take a look at the catabolic pathways for some essential amino acids: tryptophan, lysine, phenylalanine, tyrosine, leucine, and isoleucine. These pathways are crucial because they show how our bodies break down these amino acids to generate energy or produce metabolic intermediates. Each of these amino acids has a unique route through the metabolic network, but they all ultimately contribute to the cell's energy supply or biosynthetic needs. For example, tryptophan is broken down into nicotinamide adenine dinucleotide precursors, while leucine and isoleucine are branched-chain amino acids that feed into energy-producing pathways. Understanding these pathways highlights how interconnected amino acid catabolism is with overall metabolism, supporting our theme of metabolic integration and regulation. Tryptophan, as many of you know, is an essential amino acid, which means our bodies can't produce it on their own. Instead, we have to get it through our diet. But what's really fascinating is that tryptophan serves as a precursor for several important compounds in our body. For instance, it's the starting point for the synthesis of serotonin, a key neurotransmitter that influences mood, sleep, and even appetite. This connection highlights how what we eat can directly impact our mental health and overall well-being. Additionally, tryptophan is involved in producing niacin, or vitamin B3, which is vital for energy metabolism and DNA repair. So, understanding tryptophan as a precursor helps us see the bigger picture: how amino acids from our diet can be transformed into critical molecules that regulate many physiological processes. This links back to the overall theme of how nutrition influences health at a molecular level. let's take a closer look at the catabolic pathways for phenylalanine and tyrosine. These pathways are essential because they help us break down these amino acids to produce energy and important metabolic intermediates. Phenylalanine, which is an essential amino acid, is first converted into tyrosine through a hydroxylation process. From there, tyrosine can undergo further degradation through a series of reactions that ultimately produce important compounds like fumarate and acetoacetate. These molecules not only feed into the citric acid cycle, but also serve as precursors for other biosynthetic pathways. Understanding this breakdown process is key because it illustrates how amino acids are integrated into our overall metabolism, connecting protein catabolism to energy production and biosynthesis. So, this pathway highlights the interconnectedness of amino acid metabolism with our body's broader metabolic network. Although much of the breakdown or catabolism of amino acids happens in the liver, there are some exceptions. Specifically, the 3 amino acids with branched side chains, leucine, isoleucine, and valine, are mainly oxidized as fuels outside the liver. You'll find these processes occurring in muscle, adipose tissue, kidney, and brain tissue. These extrahepatic tissues have a unique enzyme called an aminotransferase, which the liver itself doesn't have. This enzyme acts on all 3 of these branched-chain amino acids to convert them into their corresponding alpha-keto acids. This is an important point because it highlights how these amino acids are handled differently from others. Instead of being primarily degraded in the liver, they are broken down in other tissues, which is a key aspect of their metabolism. So, to connect this to the overall theme, understanding where and how these amino acids are metabolized helps us better grasp the regulation of amino acid catabolism and energy production in different tissues. The branched-chain alpha-keto acid dehydrogenase complex then carries out an important step. It catalyzes the oxidative decarboxylation of all 3 alpha-keto acids. In this process, the carboxyl group is released as CoA2, and what remains is converted into an acyl-CoA derivative. Now, this reaction is actually quite similar to 2 other oxidative decarboxylation reactions we've discussed earlier in Chapter 16. One is the oxidation of pyruvate to acetyl-CoA, facilitated by the pyruvate dehydrogenase complex. The other is the oxidation of alpha-ketoglutarate to succinyl-CoA, carried out by the alpha-ketoglutarate dehydrogenase complex. What's fascinating is that all 3 enzyme complexes are structurally similar and follow essentially the same reaction mechanism. They share a common set of 5 cofactors: thiamine pyrophosphate, FAD, NAD, lipote, and coenzyme A, and the 3 proteins within each complex work together to catalyze homologous reactions. This really highlights a key evolutionary point. Enzyme machinery that initially evolved to catalyze one specific reaction was essentially borrowed by gene duplication. Over time, these duplicates evolved further to catalyze similar reactions in different metabolic pathways. It's a great example of how nature reuses and adapts existing tools for new functions, which ties back to our overall theme of enzyme versatility and evolution. Now, let's take a closer look at the catabolic pathways for the 3 branched-chain amino acids: valine, isoleucine, and leucine. These amino acids are unique because they share similar pathways for breakdown, but each also has its own specific steps. Understanding these pathways is key because they play a critical role in energy production and metabolic regulation. When these amino acids are broken down, they produce intermediates that feed into the citric acid cycle, helping generate ATP. Valine is primarily broken down into succinyl-CoA, which enters the citric acid cycle directly. Isoleucine is a bit more complex because it can be degraded into both acetyl-CoA and succinyl-CoA, giving it a dual role in energy metabolism. Leucine, on the other hand, is unique because its breakdown yields acetyl-CoA and acetoacetate, making it a strictly ketogenic amino acid. These pathways are interconnected with other metabolic processes, illustrating how amino acids contribute beyond just protein synthesis. They also serve as vital energy sources, especially during fasting or intense exercise. Recognizing these pathways helps us understand their importance in maintaining metabolic balance and how disruptions might lead to metabolic disorders. Now, let's look at how the carbon skeletons of asparagine and aspartate eventually feed into the citric acid cycle. In mammals, these skeletons enter as malate, while in bacteria, they enter as oxaloacetate. An important enzyme here is asparaginase, which catalyzes the hydrolysis of asparagine to form aspartate. From there, aspartate undergoes transamination with alpha-ketoglutarate, producing glutamate and oxaloacetate. The oxaloacetate then gets converted into malate in the cytosol and is transported into the mitochondrial matrix via the malate, alpha-ketoglutarate transporter. In bacteria, the oxaloacetate generated directly from transamination can go straight into the citric acid cycle. This pathway highlights how amino acids like asparagine and aspartate can be degraded into key cycle intermediates, linking amino acid metabolism closely with energy production. Asparagine and aspartate are two amino acids that play interesting roles in metabolism, and today I want to highlight how they are degraded into a common intermediate, oxaloacetate. Essentially, through specific enzymatic reactions, both amino acids are broken down, releasing their nitrogen groups and ultimately converting into oxaloacetate, which is a key part of the citric acid cycle. This process is important because it links amino acid metabolism directly to energy production and other central pathways in the cell. Understanding this degradation pathway helps us see how nitrogen from amino acids is managed and how carbon skeletons are funneled into energy-generating cycles. It also emphasizes the interconnectedness of different metabolic routes, amino acid catabolism feeding into the citric acid cycle. This connection is crucial for maintaining cellular energy balance and metabolic flexibility, especially during fasting or high-energy demand states. So, in summary, the degradation of asparagine and aspartate to oxaloacetate exemplifies how our body efficiently integrates amino acid breakdown with energy metabolism. This slide doesn't contain enough information. That's why we're moving to the next slide.